Howdy everyone, you're listening to the Blue Jeans and Boots podcast, where genetics grad students talk about how to navigate a PhD program while trying to stay sane. We also invite faculty from our interdisciplinary program here at Texas A&M onto the show and pick their brains about their research, what it's like being a principal investigator, and their journey to becoming a professor here at A&M. In this episode, we'll be talking to Dr. Zach Adelman. Dr. Adelman is a professor of entomology here at Texas A&M with a special focus on the genetic control of the yellow fever mosquito Aedes aegypti. My name is Joe. I'll be one of your hosts today.
And my name is Serina. I'll be your other host today.
All right.
Welcome to the show, Dr. Adelman.
Thank you. I am very, very excited to be here.
We're excited to have you. How is your day going so far? It's Friday.
It's Friday, and it's not quite hot yet, so I was able to walk over here no problem.
Yeah, it's definitely getting toastier, which is unfortunate.
It's the Texas heat. Hopefully you're used to it by now.
I am. I know what to expect. I don't know that you.
How long have you been here, Dr. Adelman?
7 years now.
That's exciting. You're coming up on a decade. That's a big deal.
Yes, it used to be consistently like 96, 97 every single day, and now it's 102, 103 every day. So things have gotten hotter since I've been here.
Especially in July and August.
Yeah.
It gets pretty rough. So before Texas A&M, where were you and what brought you to Texas A&M?
Before Texas A&M, I was associate professor and assistant professor at Virginia Tech. So this is in Blacksburg, Virginia. I got there in 2005 and was there until 2016. So I was there 11 years before coming here.
Wow. Does it get as hot there as it gets here?
It does get as hot.
Oh, wow.
But not for as long. And this is what I try to convince people that are in the north, that it gets just as hot in Virginia or New York as it does in Texas, but for like a week or 4 weeks, 5 weeks, not for 4 months.
Yeah.
That's the big difference. It's not that it's hotter down here. It's just that it never ends.
It's just hotter longer.
Yes. Yeah, I explain the same thing to people who have lived like either in Texas or neighboring areas, that it's just as humid.
Yeah.
Back in the Northeast, but we have 8 months of cold, not 8 months of heat. So yeah,
it's a different kind of climate.
Yeah, definitely.
But what brought you to Texas from Virginia?
We drove, so my car.
Excellent. What was the motivation for you to drive your car from Virginia to Texas, Dr. Adelman?
Well, I had a job lined up here, and so that's what brought me here. But no, I was in a position where I felt like there were things I wanted to do that I wasn't able to do, and talking with folks here in entomology in Texas gave me the opportunity to do some things that I was not able to do at Virginia. And so I thought it was a good time to move, and it's certainly been good for me, and it's certainly been good for my family. I think, family reasons, this was a really good schools here and a lot of things for kids to do, and that was important for me as well.
Okay.
That makes sense.
I guess piggybacking right off of that, speaking of things that you wanted to do coming to Texas A&M, can you talk a little bit about your current research and the different projects that are going on in your lab?
Sure. So as you mentioned in the intro, my lab works with the mosquito, dengue mosquito, yellow fever mosquito, Aedes aegypti, and we use gene editing technologies to understand the genetic basis of different behaviors and then try to use genome engineering to find new ways to control this vector. And so we have a lot of projects that are based on either genome editing or genome engineering, bioinformatics, genomics, trying to tie different genotypes to phenotypes, and then again using biotechnology to kind of create new phenotypes that might benefit us, like mosquitoes that can't blood feed or die very soon, or all these kinds of things that we'd like them to do to stop transmitting viruses.
Can you talk a little bit about how you go about making mosquitoes, say, that die sooner or don't blood feed or anything like that?
Sure, trying to identify genes that are important in those processes and knock them out. And so if it's different genes, if you knock them out, the mosquito, rather than being able to digest blood, chokes on it and would die. Ooh. Or mosquitoes that, they've lost a gene that's involved in flight, and so they just dance around helplessly on the bottom of the cage, and you can just taunt them because they can't get you, right? So these are things we've done recently. Dr.
Adelman wants to create a world where we can taunt mosquitoes.
You just put it on the counter and say, "Come get me," and then it's just kind of stuck going, "I'm thirsty, but I can't fly."
Just a little bit of bug torture, tiny bit.
Well, that's how I got into mosquitoes in the first place.
Bug torture?
Mm-hmm.
Or human torture? I mean, they torture us.
So when I wanted to go to graduate school, I knew that I wanted to work with viruses, but I didn't want to work with animals because I didn't want to. And this is nothing against using animals in research. I just knew that I couldn't do it.
Yeah.
But I had no problem pulling the legs and wings off a mosquito. That was no problem for me. And so there was a career waiting for me there.
Awesome. Yeah. Mosquito torture.
So, I guess related to the mosquito research, how prevalent are the diseases that mosquitoes are responsible for in the United States and in other parts of the world?
So I'll sidetrack your question a little bit before answering it, because Joe knows about my love and hate relationship with the word mosquito, because it's a very confusing term, right? And so when you think of mosquito, you think of a small thing that buzzes around and sucks your blood, right? And so that's a pretty accurate definition of a mosquito. But it loses the nuance that there are more than 3,000 species of mosquito that are evolutionarily extraordinarily distinct from each other. And they have different genetics, different biologies, different ecologies. Many of them are not vectors of anything. Many of them don't bother people at all, but a few species do. And so, in terms of malaria mosquitoes, which are 200 million years or more separated from the dengue mosquitoes I work with, you have malaria, which has 250 million cases per year at this time, but the yellow fever mosquito, we're talking about dengue cases of, hundreds of thousands to millions of cases, depending on whether or not you're talking about highly symptomatic cases or just any cases. And then, of course, billions of people at risk that live in these areas.
That is insane to me that there are so many different kinds of mosquitoes and that we only interface with like a very small subsection of so many species of them.
Yes.
That's crazy. I don't know, sometimes I forget about the nuance of like specialized biology versus like the wholeness of the ecosystem that it operates in, which is insane.
Well, it's hard because they're small and they're all the same size, right? So you take a malaria mosquito and a dengue mosquito, you put them next to each other, and someone would say, well, those are just two mosquitoes. But if I put you next to a duck-billed platypus, where you are more related to that platypus than those two mosquitoes are to each other, right? It's obvious that you guys are not the same thing.
We're nearly identical.
You're nearly identical. Yeah, exactly.
Wow. That's an excellent analogy. I wouldn't have thought about it that way, but that's a good point. Yeah, that's a good point.
So I think we touched on it before a little bit, but what do you think you'd like to ultimately solve with your research? Because there's a lot of different projects that are going on in the lab, but what's like the main, I guess, motivation?
Well, our lab is motivated to kind of create useful information and useful tools. And our hope is that we will actually get something out of the lab and into the field to be used as a useful tool. But we would like to be responsible in our development of technology as well. And so this is a two-edged sword. We have people whose lives are constantly at risk of these viruses, but we are not interested in pushing something out the door that we don't believe is safe to use in terms of biotechnologies. And so a lot of time is spent in the lab on research on how to control technologies that are developed in the lab and how to evaluate them for safety. And so we can't be in a rush. We have to develop these things responsibly. And so my hope is that the information we identify, what we learn about these mosquitoes and these genes can be used eventually, hopefully soon, but that's not the primary goal, is to rush something out the door, because I don't think we can do it responsibly if we did that.
What is your definition of soon? Like, how soon do you think it could happen, theoretically?
So I answered that question for a reporter as a postdoc. My PI had just published a paper on, some mosquitoes that were modified to contain an antibody that recognized a malaria parasite.
Oh, wow.
And he wasn't around that day. I think he was traveling, and a reporter called the lab and said, when do you guys think those are going to be ready for the field? And I said, 2 or 3 years. This is, of course, in 2001, right?
Oh, wow.
And those things have still not been in the field yet. So there are so many things that we have to learn about these mosquitoes, and there's so many things you have to communicate to people who are not familiar with this technology and need to know about it, and either are skeptical about it or just have real questions that need answers about what happens at this, what happens at this, what happens with this. And now you're navigating the regulatory system, you're navigating what people want, and of course the problem is changing as you're navigating that pipeline.
Right.
So I do not give answers to that question anymore because I honestly don't know. But people are pushing these technologies forward. We're not the only lab that's doing this. There are other people pushing these biotechnologies out that are different stages of development, and the world will get them little by little as they are evaluated for safety, and they will make their way into the field, but it will happen in due course.
Yeah, gradually. You bring up the regulatory aspect of doing things like genome editing and even like making things to release sort of into the wild someday. And it's interesting because a lot of the regulatory framework that exists around like animal usage or human usage in studies has to do with public response to studies that had already been done, and that's where a lot of the regulation came from. So if you could tell the general public something to sort of encourage them that this technology, assuming someday it does get released, would be helpful, what would you want people to know?
Oh, I definitely won't answer that question either.
Okay.
Because
Fair.
I can't, every technology is different, and some uses of it I might be opposed to and would say this probably isn't safe to use.
Okay.
Other instances, based on what it looks like, I would say it definitely is. And so it depends on which technology we're talking about. But I definitely wouldn't say blanket biotechnology good. Things can be used responsibly or irresponsibly.
Yeah.
So you talked a little bit about biotechnology and responsible use of the biotechnology to control these Aedes aegypti populations or malaria mosquito populations. Just for our listeners, because I'm not sure if anybody if there's any mosquito experts that are out there, but could you talk a little bit about some of those different approaches to controlling them and the biotechnology that's associated with it?
Sure. So, in general, we talk about two different approaches. One is based on suppressing the population, right? So making mosquitoes that are essentially males that can't produce fertile progeny. So a male mates with a female, and the female then either doesn't produce any offspring or only produces male offspring. Male mosquitoes do not feed on blood. They do not transmit pathogens. And so having male mosquitoes, people wouldn't really notice them at all. And of course, eventually you run out of female mosquitoes, either because none of their eggs hatch or because they're only producing males, and that process continues. And so eventually the population would dwindle down and decrease to the point where transmission would be interrupted, right? So these population suppression approaches, and this is happening with Aedes aegypti. If you're talking about incompatible insect technique, if you're talking about the Oxitec mosquitoes that are being released in Florida, these are all sterile insect technique-based processes that are currently being used in the United States to reduce mosquito populations, to success from what I can see. And similar approaches are being developed by others for malaria mosquitoes. And then there's the other half of the coin, which is the population replacement approaches. That is, rather than reducing mosquito populations, which sounds great, but then if you ever stop, the mosquitoes come right back again. And so you basically have to release these things until the end of time to keep those mosquito populations down. So it's a lot of cost and a lot of effort to do that. And as we see in the US with mosquito control, when you have West Nile or you have Zika, outbreak, big pot of money, outbreak goes away, all money disappears, and then new outbreak comes because there's no ability to detect it, all the surveillance is gone, all this infrastructure is broken down, and then outbreak, and then new pot of money, right? So we don't sustain things. Nobody sustains things. When there's no cases, the money goes away, and then programs dry up, and then things come back again. So the alternative then is these population conversion approaches, where you take a population of mosquitoes that's very good at transmitting viruses or parasites and modify their genome so that they are no longer good at transmitting viruses or parasites. They'll still bite you, they'll still annoy you, but they won't kill your children, right? That's the idea behind those approaches. And now, once you've converted them, then they just stay like that, and you don't have to do anything more is the goal. So those might be more sustainable, so you live with mosquitoes, but then you get rid of the transmission.
That's a good summary of a lot of different approaches there.
Yeah,
it's a mouthful
honestly.
for sure.
Can I ask further? So I am not a mosquito person. I'm not terribly familiar with everything that's going on in the mosquito world. What is going on in Florida? Are they releasing sterile mosquitoes?
So they are not, by the definition of sterility, they are not sterile.
Okay.
But the transgene that is going to be inherited by the progeny is going to kill them as larvae. And so they don't grow up to become adults that are also fertile. So it meets the same type of end result. That is, the females don't produce any viable female offspring, but they will produce viable male offspring.
Okay.
And so those male offspring can come out, half of them will inherit the transgene and can pass that on again to kill more female offspring. And so you have kind of multi-generational for a few generations, but it still does go away, and it's still based on killing rather than conversion.
That makes sense.
So we've talked a lot about some of the solutions that have been proposed in terms of like genetic control of the Aedes aegypti mosquito and other mosquitoes like Anopheles mosquitoes. But these are more like logistical or technical solutions. And as we know that not every problem is just going to be a technical problem, I think one thing that's really interesting about the vector control field is that these issues are usually multi-layered. So could you talk a little bit about some of the issues that surround the control?
Sure. I mean, I work in the field of mosquito genetics and biotechnology, so this is where our focus is, but there are still plenty of people working on improving drug development, improving vaccine technologies, working on improved bed nets, longer lasting bed nets, indoor residual spraying, community programs to remove breeding sites where possible, especially for dengue mosquitoes. And awareness of mosquitoes and pathogens and how to keep yourself protected, right? So all of these things are going on, and people specialize in all of these things, and that needs to continue, right? This is one additional tool for the toolbox, not a replacement catch-all to get rid of those other things, right? That all those things are going to still be around.
Makes sense. Maybe a ridiculous question, but from a plant scientist standpoint, I was thinking about biotechnology, and usually for us there's two sides. You can use biotechnology to solve a problem, or you can use breeding to solve a problem. Does anybody do mosquito breeding? Like, is anybody trying to, I guess that's kind of like the killing the larvae thing, but is there like a subset of mosquitoes, like the ones that don't bother us, that could cross with the ones that do?
A long time ago, yes, there was a thought. So people kind of came across this through conventional breeding, where they found some strains of mosquito are more refractory to virus transmission or more refractory to parasite transmission than others. And the idea was, can you breed up a population from this that could do that? And people tried for a while to do that, but it ended up being more challenging than they thought. That is, there are mosquitoes, if you're refractory to dengue 2, this strain, it turns out that's not universal. You might be highly susceptible to a different dengue strain.
Okay.
And then mosquitoes from this region that were found to be highly resistant to that strain are highly susceptible to these other 3 strains. And so universal resistance was eventually not discovered. And so people kind of gave up on that, finding something that was naturally resistant to all possible viruses, right? Rather than just the one that they grew up with. They're resistant to that one, but then a new one is, and because dengue viruses move around the globe so much, they're constantly being seeded in new places. We've kind of moved away from that towards these other biotechnologies, but maybe there's enough genetic diversity in Aedes aegypti to do that, maybe not, I'm not sure.
Okay. It's interesting.
That's a good question.
Yeah. It's curious.
Mm-hmm.
So we talked a little bit about the responsible gene editing, and with that comes regulation. And I am aware that I believe you're involved a little bit in some of the different government organizations that are bringing awareness to responsible use in gene editing technologies and biotechnology. Could you talk a little bit about that and maybe what those regulations look like current day?
Sure. So you're talking about in the United States?
I guess in the United States and everywhere, that would be.
Well, certainly I can't speak to everywhere because every country's got their own structure for how they regulate biotechnologies. And I can speak a little bit about what is happening in the United States in terms of mosquitoes.
Sure.
And this is at the present time based on guidance put out by the Food and Drug Administration a few years ago. Basically assigning, they have this joint regulatory framework where the USDA, FDA, and EPA kind of get together and decide who's going to take the lead on regulating a different product based on its intended use. And so the current guidance is that if your goal is to kill mosquitoes using genetics, using a pesticide, whatever it is, then the EPA will be the lead agency that regulates that. And so the Oxitec trials that are going in Florida are regulated through the EPA. Incompatible insect technique that uses the Wolbachia bacteria goes through EPA because the idea is to produce fewer mosquitoes. And that is the endpoint that they are going to be measuring for those permitting. If your goal is to reduce disease transmission in humans, and that's your stated end product that your product does, then the FDA will be your regulatory agency. And so that could be these conversion ones where you're leaving the mosquitoes in place and just making them so there's less disease. Right?
Wow. It's interesting. I didn't know that it was endpoint regulated. Like that was how they divvied up.
Yes, it's based on the
what.
claim that you're making with your product.
Okay, interesting. Huh. It's weird to think about science becoming a product. Sometimes we spend so much time in the lab like working on the nitty-gritty pieces of something.
Yeah.
So then when it becomes a product out in the world that like you have to, I imagine like marketing like, oh, this is a good idea and this is going to be good for people, and that's a whole separate thing that people have to worry about, which is crazy.
Yeah.
So, I guess we'll zoom out from the research part of your job, and let's talk about just being a professor. So what made you want to be a professor?
Let's see. I enjoyed doing research when I was an undergraduate, and I did not like, what's the way to phrase this? I have a problem with authority, and I wanted to do things my own way. And if you want to do things your own way, there's basically only one choice. You gotta be in charge, right? And so that's what led me to combine those two things together.
Awesome.
That was very punk.
I have a problem with authority to be the title of this episode. I have a problem with authority with Dr. Zach Adelman.
Yeah, that's our soundbite.
Yeah, there you go.
That's it. It was perfect. Did you know you wanted to work with insects in undergrad? Did you start with insects?
No, I didn't. I worked with frogs as an undergraduate.
Oh, really?
Yeah, I was in a Xenopus laevis developmental biology lab. So, we would get frog embryos at different stages, dissect them, extract RNA. I had a cDNA library that I used to screen, using filters in P32 and whatnot. So
Ooh.
old time manual sequencing. We can go back to the dinosaur age for my undergraduate here. But no, the mosquito thing came about because, as I said, I was looking for virology labs that did not work with mice. And that actually was really challenging because almost all of them were focused on viruses that affect humans. And those were using mouse models. And so the mosquitoes came about because they were the model system. I was interested in studying the viruses first. But then after working with mosquitoes, I just enjoyed that a lot and went to the mosquito genetics side primarily.
Mm-hmm. That makes sense. What got you interested in viruses specifically?
I don't know. As an undergraduate, they were just super cool. They have almost no genes. They're super tiny, and then they can cause all these big problems. So
that's also a very punk answer.
Yeah.
Well, and technically, they're not considered living still, are they? I worked on viruses briefly in undergrad, and I remember that confused me.
Yes, I do not consider them living.
Okay.
I consider them a set of instructions that are wrapped in a package, and it's up to the cell to open the package and either follow the instructions or not.
Scary.
Famous Dr. Adelman analogy. If you take his classes, you'll know.
It's like when people send glitter in the mail. Have you heard about people doing that?
I have not.
You can pay to have someone send like mail an envelope full of glitter to someone. I think
just to mess with them.
Yeah, just to mess with them. The virus is the envelope full of glitter. The cell opens it up and it's like, oh no, now I have a mess.
We're going to start delivering glitter to cells.
No, but it's even worse than that, right? Because it's a recipe for how to put glitter in envelopes that you put in the envelope so that the other person does it and sends it to a bunch of their friends.
It's like a Black Mirror episode.
Yeah.
Okay, so I have a quick question. So you brought up being interested in the virology side of working with mosquitoes and the Aedes aegypti mosquito in particular. So it seems like you're taking you took more of a microbiology approach, I guess, to
my PhD was in microbiology.
Yeah. So in vector control, which is what we'd call the control of these mosquitoes that spread pathogens that cause disease and controlling those populations, there's a lot of different backgrounds, I think, that are brought to the field. So I know that you have some historical context of the beginning of that field of controlling the mosquitoes. I was hoping maybe you could elaborate a little bit on that.
On how long this has been going on or discussed, or what fields get brought in?
I guess what fields get brought in, yeah.
Pretty much everybody.
Yeah.
I mean, sociology, ethics, psychology, political science, ecology, landscape ecology, modeling, right? Everybody is involved and can contribute to this.
If you don't think you're involved with mosquitoes, let me tell you, you are.
But everybody's affected by mosquitoes, right? Nobody says, mosquito, I've never heard of it. I've never gotten that from anybody.
That's true.
Right? I tell people I work with mosquitoes, and they're like, oh, I hate those, right? Or, can you kill them all faster, right?
Yeah.
I never get somebody who says, can you explain what a mosquito is to me? That has not yet happened, no matter where I am in the world, right? So.
Although you gave a very good explanation of how a mosquito is a confusing term, and I appreciated that.
Yes.
So I have taken a class taught by you, and I'm curious, what is the favorite class that you've ever taught?
Yeah, parents don't usually pick and choose their favorite children's. So.
Not my class in particular, but the actual course, I guess I'm asking.
So for me, it's about just the interactions with the students, and that is a organic event that happens, and the class takes on a life of its own beyond the material. And so I enjoy every moment that I'm in the classroom, and I'm not going to pick a favorite child. And I don't want a student to enroll in my class and say, well, this is your second favorite class. We're getting the substandard. When I'm in the classroom with the students, that's where I am. And then we talk about all kinds of fun things.
Every class is your favorite class.
They are, because why am I doing it if I'm not giving what I have to that class, right?
That's
and
true.
so there is no second class. It's you come in and you have fun with it, and you go take the students on an adventure.
Do you teach undergraduates and graduate students?
Yes, I have 2 classes coming up this fall. One is a split, so it's both undergraduates. I think I have 11 currently signed up, and then there's 3 graduate students in it, and then the second one is dedicated to graduate students.
Very cool. Do you feel like you have to take very different approaches with your graduate classes versus your undergraduate classes, or are they both, do you treat them relatively similarly?
They're relatively similar. I think that, there are different demands on the time of an undergraduate and different demands on the time of a graduate student, so the types of projects I ask them to do might be a little bit different. But in the classroom, what I've seen is that the undergraduate students do just as well as the graduate students, and that's not a knock on the graduate students. It's just that, I think it's new for everybody, the material that I cover.
Right.
When we go into deep depths on these mosquito control programs, it's not like a graduate student has had a lot of experience in that either. And so everybody's kind of coming in from square one. Everybody knows about mosquitoes. People kind of have their own opinions of them. And then we talk about these technologies, and it's kind of new for everybody.
Yeah. That's cool.
So a question about being a professor and mentoring graduate students. Is there any advice that you would give to incoming graduate students that are, just about to start their PhD or master's program?
Buckle up. It's going to be a bumpy ride.
That's very fair advice. Accurate so far.
Persevere.
Accurate. Yeah, that's true. I tell a lot of people that you don't have to be, I think you need to be smart to get a PhD. You got to be able to do the work and understand the concepts and pass your prelims, pretty important. But really, above all else, you need to be stubborn.
Mm-hmm.
You have to be really, really, really sure you want to do this.
Well, I think you guys have heard me give the analogy of the escalator or stairs versus the rock climbing, right? And I'll go over this in the orientation, but graduate school is the equivalent of, bouldering or rock climbing, right?
Okay.
You can go up, but if you don't make it to the top, you're coming back down. And then you got to start again and find a new route to go up. And so you can spend years climbing, but not reach the top, and you're at square one where you started from. And so a lot of graduate students get discouraged because they're at year 2 or year 3, and they just don't feel like they've gotten any closer to the end, right? But they've gotten much better at climbing because their hands are more callous, they've gotten which route to take, and so eventually they'll just scramble right up to the top like it was nothing. And then if I show them a new wall, they can just scurry up to the top of that one too, right? But it's not going to climb itself. You've got to go up, fail, go up and fail a lot of times to get going. It's not like undergraduate at all, and that mindset takes a long time to break somebody out of that. Look, you're not just going to do one semester and be one semester closer to finishing.
Right.
It's not the way it works at all.
Yeah. That makes sense.
Another famous Dr.
Yes.
Adelman analogy.
Yes. New students in orientation will hear that again in a few weeks.
You'll be able to tell who listens to the podcast by who already knows what's going on.
Right. That's right.
So for our new students and for our incoming students, what skills and qualities do you look for when mentoring students? and how do you think you foster their professional development and success? I feel like this is an interesting question for me to ask, being your student, but I'm asking it. So
why don't I say, Joe, how have I done so far? You can go ahead and
You've done a good job. Yeah, you've done a good job.
What do I look for? I look for students that are passionate about being in that environment, in that lab, students that come in wanting to accomplish something, because I can't give that to you. I can give you a lot of skills on how to design experiments, how to evaluate literature, but I can't give you the passion to want to do science, right? And so somebody's going to have that already, and then there's a lot you can do to work with that. But if somebody's like, I don't even know why I'm here, there's not a lot I can do.
I think Serina was hinting at it before too, and you were as well with your answer with advice to graduate students. But in a PhD, there'll be a lot of failed experiments that'll really test whether or not you want to be here and how stubborn you are. Yes. So absolutely. I think you need to have that motivation, that desire.
I mean, I think the primary way I'm supporting students, and the beginning students probably don't like this at all. The advanced students appreciate it tremendously. Is that I don't tell you what to do. I ask you to come up with a plan. He comes into my office with a problem. Here's the problem. Okay, what's your plan for fixing it? And I want to hear his plan or her plan, and then I'll say, okay, that's your plan. This may not work in that plan, or we can change this in this plan. I critique a plan, but I don't just give a laundry list of do this next, do this next. That's not doing the student any favors.
Yeah, I guess from a student's perspective, being in Dr. Adelman's lab and being my mentor, that's one thing I cannot stand, is being micromanaged, and he has not done that. He's always given me, just enough leash to go and run off, but then I kind of get pulled back when something goes wrong, and I'm in his office like, hey, this isn't going right. Show him some gel or some sequencing results, and he's like, well, why isn't that going right? Let's talk about that. So, I absolutely believe in that philosophy too, and I appreciate that.
You don't tell people what to do, and you have a problem with authority. You're not trying to enforce authority. I appreciate that.
Well, I would like people to be independent thinkers, and they have to achieve their own destinies, and that's how you cultivate that, is you give space without letting them just wander forever, right? And they need to explain to me what they're doing and why, but I'm not going to tell them what to do.
Makes sense. Well, I think we're just about wrapping up. Is there any final thing you want to leave our audience with today, Dr. Adelman, about your work, about mosquitoes, about graduate school? Anything you want?
They'll just have to come and talk to me and find out.
Okay, awesome.
A cliffhanger.
A cliffhanger. Awesome.
All right. Do you want to take the outro?
Sure. Today's episode was hosted by Joe Romanowski and Serina DeSalvio and produced by Anthony Matarazzo. This podcast is brought to you by the Texas A&M University Interdisciplinary Graduate Program in Genetics and Genomics. If you have questions about our program here at A&M, please feel free to email us at bluejeansandboots@gmail.com. You can also find us on Facebook, Instagram, Twitter, and TikTok at Genetics Podcast. Thank you for listening, and we'll see you next time.